Hello students, how are you all? Good evening. I hope you all are doing great. So as we are continuing with the chapter Cell, the unit of life, which is a very important and interesting chapter, and in this we have covered all the prokaryotic cell organelle and we are discussing the cell organelle present in eukaryote. So here the last cell organelle we have studied is the centrosome. So this was the last cell organelle which we have studied. So here we have seen that flagella or cilia is present on the surface of they are hair-like structure present on the surface, right? And here we have said that axoneme is the exposed part and basal body is present inside the membrane, right? It is present beneath the membrane, and then we have said that axoneme has 9+2 arrangement and basal body has 9+0 arrangement. Here we have already seen that if this is the extension of membrane, then the below part will be the basal body and this part will be the axoneme. So axoneme is the exposed part, right? And this part is the basal body. So in last class we have already discussed in detail about the axoneme. Now today we will focus on the basal body, but before going to the details of basal body, just once again we will see the arrangement of axoneme. So first of all, when I say arrangement of axoneme, what arrangement we are talking about? Arrangement of what? So the arrangement of microtubule. When I say 9+2 arrangement or 9+0 arrangement, this is the arrangement of microtubule, right? Microtubules arrangement. So let's see here, this we have already discussed that there is this 9+2 arrangement where 9 doublets are there, right? Now if I ask you, these doublets are connected by, so these you can call it as linkers. So how many linkers? So here let me summarize this point. Here if I ask you, and you all can write it down. So here if I say peripheral microtubule, tell me the number. Peripheral microtubule means microtubule which is present at the periphery. So peripheral microtubule is 18, right? How 18? 9 doublets are there, so 9 into 2, 18. Then comes the central microtubule. So central microtubule, there is 2. You can see here 1, 2. But they both are not attached to each other, so we don't call them doublet. They are singlet. So 2. So if I ask you total microtubule, so you will say total microtubule is 20. Total microtubule is 20. So I think this part till here we have also discussed in the last class. Now apart from these numbers, sometime question is asked about how many linkers. How many linkers? Now here if we talk about linkers, so see in both this case here doublets are connected through linker. So 9 doublets are there. So how many linker will be here? 9 linker in the periphery, and 1 linker is also at the center. Understood or not? There are 9 doublets and in circle they are connected. So 9 doublets will have 9 peripheral linkers and 1 central linker. So total linker, can we say how many linkers? So we will say 9 plus 1, that is 10 linkers. So if question is asked how many linkers, so you will say 10. 9 peripheral and 1 central. Fine. Then next question, this one we call it as the radial spoke, right? This one which is coming out from each doublet, you can see one spoke like structure is coming out. So this is called radial spoke. So if we mark it, see, 1 radial spoke, 2 radial spoke, 3, 4, 5, 6, 7, 8 and 9. 9 doublet, 9 radial spoke. So number of radial spoke will be 9, right? Number of radial spoke is 9. So this is all about the axoneme, which is 9 plus 2 arrangement. All question can be answered if you can keep these things in mind. Now talking about the centrosome. So here two terms are there. One is called the centrosome, another is called the centriole. So let's first understand what is centrosome, where is it found. So centrosome, if you all can recall cell division, which you must have studied in class 10th, right? So when you see the cell division, there inside the cell you see spindle is formed. So here on the two poles, what is present? It is the centrosome which is present, through which the spindle apparatus is formed. See, spindle apparatus is present. Is it clear? This diagram all of you can easily recall, right? This is a cell division, and here of course chromosome will be there. In the next class we will discuss about that, but this we will call as the centrosome. So how many centrosome are there? 2 centrosome are there in a dividing cell, right? 2 centrosome represent 2 poles. But then what is there in the centrosome? How a centrosome look like? And how is centrosome related to centriole? So let's see that question. If we say centrosome, so centrosome consists of 2 cylindrical centriole. Centrosome consists of 2 cylindrical centriole like this, and it has what angle? 90-degree angle. Means 2 centriole perpendicular to each other form a centrosome. So can we say that centrosome has 2 centriole? 2 centriole. Each centrosome has 2 centriole, and a dividing cell has 2 centrosome. So 2 centrosome means how many centriole? How many centriole? So there are 4 centriole, right? In a dividing cell. Now question comes, this in every cell there are 2 centrosome? So answer is no. In normal cell, in a non-dividing cell, there is only 1 centrosome. Remember this point. In a non-dividing cell, there is only 1 centrosome. But if it is a dividing cell, then there will be 2 centrosome. Means what? When the cell will divide, centrosome will make a copy of itself, right? In the next chapter we will study. Now it is a question. If anyone can tell me, then I will take the name of the student. If I ask you, and this is the challenge question, right? So all of you listen it carefully. This is a challenge question. So if I give you this question, centrosome duplication take place in which stage of cell division? Tell me, in which stage of cell division the centrosome duplication take place? Yes. Those who will answer correctly, first person who will answer correctly, I will write the name and give star. Let's see who can answer it. In which stage in which stage centrosome duplication take place. See, usually there is only one centrosome in the cell. When the cells decide to divide, the centrosome become 2. In which stage centrosome duplication take place? Okay, Sai has said that S phase, Srinivas has said S phase, Sanmukh S phase, okay, Faz also says S phase, okay, Pinky S phase, Tanu S phase, Jaideep S phase, Tanu, Sharanya, very good. So yes, so just those who have answered, 5 star to you. All of you who have answered S phase, right? 5 star to you. Now here, see, simple. Here, centrosome duplication take place in S phase. S phase, right? So all student who have given S phase, it is correct answer. So you got 5 star. Now those who were unable to figure out, don't worry, we haven't covered it, right? This is a part of next chapter. In next chapter we will see what are the different stage of cell cycle, and there we will see what happened in S phase, what happened in G phase, etc. So from here what we will, if we summarize this entire part, so first thing, what we will say, centrosome consists of how many centriole? So it consists of 2 centriole. These centriole are perpendicular to each other, means 90 degree. A dividing cell has how many centrosome? A dividing cell has 2 centrosome. A non-dividing cell has 1 centrosome. Centrosome duplication take place in which stage? So it take place in the S phase. So can we say that centrosome help in cell division? Yes. So now overall we concluded that a centrosome has centriole. So now what is centriole? Each centrosome has 2 centriole. Then what is centriole? So here, see, this is overall both centriole together form the centrosome. And now you can see these 2 centriole which are 90 degree to each other. Now if you observe them carefully, these are the microtubules present, right? You can see the microtubule arrangement. So these centrosomes are also made up of microtubule. What arrangement of microtubule? So I have told you earlier also that in the centriole the arrangement is 9+0. Microtubule arrangement is 9+0. Now, if question is asked, what is the arrangement of microtubule in, what is the arrangement of microtubule in centrosome? Still you will say 9+0. If it is asked in centriole, still you will say 9+0. Because centrosome is made up of centriole and centriole has 9+0 arrangement. This point is clear. So now we will see that what is this 9+0 arrangement. Let's see that part in detail. So here you can see this. Now if you observe it very carefully, so this is the 3-dimensional diagram. Let me first try to explain this and then I will come to this 2-dimensional diagram. If you observe this, again, isn't it a cylindrical structure? First of all, if I simply simplify it, then this is a cylindrical structure. Now you see here 1 microtubule, 2 microtubule, 3 microtubule. Again 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. So now can we say that 3 microtubules are attached to each other and these 3 microtubules are forming triplet? In 9+0 arrangement, there are triplet of microtubule present. In 9+2 arrangement, doublets were present. If you can recall here, see, here doublets are present, which I was drawing like this. But if it is triplet, it will be 3 microtubule attached together. So in 9 plus 0 arrangement, there are triplets present, right? Now let's see this two-dimensional diagram. Here it is more clear. If we see here, see, triplet. Now if I, just for the sake of simplicity, if I do the numbering, let's say this one I will call A, B and C. Again this one I will say A, B and C. A, B and C. A, B and C. So if the triplet consists of A, B and C, so now can I say C of every triplet is connected to A? C is connected to A, C is connected to A, C is connected to A. So that's why there is a CA linker or simply linker. Can we say that? So now if you see how many triplets are present, so if you count again 9, 1, 2, 3, so I will write 1, 2, 3, 4, 5, 6, 7, 8 and 9. So how many triplets? 9 triplets are there. So first of all, 9 plus 0 arrangement, in that what is 9? So 9 is the peripheral triplet. Fine, peripheral triplet. Now 0 means this second position represent the central position. So 0 means there is no you can clearly see there is no microtubule in the center, although circular appearance is there, but this is not a microtubule, this is called hub. Although hub is also made up of protein. Hub is also made up of protein. Now, so central hub is there, and see, from the hub is coming out this radial spoke, right? You can see radial spoke. So how many radial spoke? 1, 2, 3, 4, 5, 6, 7, 8, 9. So again 9 radial spoke is there. No central microtubule. So here if I have to write, here also there is peripheral microtubule. Peripheral microtubule. Now tell me, how many peripheral microtubule? How many peripheral microtubule? Star question. Star question. Let me see how many of you can answer it. How many peripheral microtubule? How many peripheral microtubule? Okay, Pinky, Khalil telling 9. Okay, see, 9 triplet, yes, Tanish has given correct answer. See, 9 triplets are there, microtubule. Each triplet is having 3 microtubule. Yes, Sai has given, Jha has given correct. So each triplet has 3 microtubule and the total 9. So number of triplet will be 9 into 3, that is 27, right? So 27 is the peripheral microtubule. In case of 9 plus 2 arrangement, we have seen 9 into 2, 18. Now here if I ask you next question, again you answer. This is also a star question, central microtubule. Now this again just I have said just earlier, central microtubule how many? Now for central microtubule tell me. Central microtubule, yes, again Tanish has given correct answer first. Okay, Ishwar, Jha, Tanish, very good. Ishwar is giving right answer. Chandrika correct. Avnish. So many, Raju, Faz, Sai, fine. So central microtubule, it is clearly mentioned here. 0 is the central microtubule. So no central microtubule present. Instead, in the center what is present? In the center is present the hub. And from the hub is the spoke, right? Now, if you see this structure, isn't it looking like a cartwheel? Such is the cartwheel structure, not drawn properly. Cartwheel appears something like this, no? Let me draw it properly. So, cartwheel means bullock cart. There is bullock cart wheel look like this. There is a central hub and then there is a spoke, right? This is cartwheel. Bullock cart look like this. So, that's why it is called a hub, central hub and spoke. You can also correlate with the bicycle. In the bicycle you have a spoke, and in the center all the spokes are connected to a hub, central hub. So hub and spoke together they form cartwheel like structure. So question if asked, cartwheel like structure has what arrangement? You will say 9+0 arrangement. Cartwheel like structure is present in, you will say, centriole. So this point is totally clear. So if these 2 points are totally clear, then one more point you have to understand, that centriole form the basal body. Centriole form the basal body. Basal body of flagella or cilia. So if now I ask you, basal body of flagella or cilia has what arrangement of microtubule? What you will say? Basal body of cilia and flagella has what arrangement? What you will say? It has a 9+0 arrangement. And if I say axoneme of cilia and flagella, so you will say 9+2 arrangement, which we have discussed here. See? Basal body has 9+0 arrangement and the axoneme has 9+2 arrangement. What is axoneme? What is basal body? Already explained. So now let's move ahead. So now if overall, so we have this understood this. Now here you also participate with me, right? So so that we can write some important characteristic of both. So if we say 9+2 arrangement, what has 9+2 arrangement? What has 9+2 arrangement? So I will say axoneme has 9+2 arrangement, whereas the basal body or centriole has the 9+0 arrangement. Simple. Second point, in 9+2 there is peripheral doublet or singlet. So peripheral doublet is present, and here present peripheral triplet. Now here if I say central singlet, central singlet, and here central absent. Here central is absent. Is this point clear? Then further if I say total microtubule here is, tell me answer. Total microtubule here is how many? Tell me total microtubule in 9+2. Quickly, fast. Let me see who can answer first. Total microtubule in 9+0 and total in 9+. Tell me. First tell me for total microtubule in 9+2. Yes. Okay, Chandrika, Chetna, Geeta, very good. So you are telling 20, very good. So 18 are the peripheral and 2 are central. So it will become 20. Now here total microtubule will be, so here simply 27 peripheral and 0 central. So total will be 27. This point clear? Now, radial spoke. Radial spoke. How many here? Tell me. Radial spoke. Radial spoke in 9+2 arrangement is radial spoke. Yes. So all of you, those who are answering 27 is correct for the total microtubule. Very good. But now I am asking here about radial spoke. Tell me, how many radial spoke in 9+2 arrangement? Okay. L. Chandrika is telling 9. Okay, very good. Chandrika is telling 9. Okay. Sheikh is telling 9. Geeta is telling 9. Khalil is telling 9. Utkarsh 9. Fine. So here radial spoke will be 9. Some of you have written 10 also, right? So I will show you. See, radial spoke in both the case is 9. Just let's see once. Let's see, go back and see here. So here we have just counted, right? It is 9. 1, 2, 3, 4, 5. So radial spoke is 9. And here also, if you count, radial spoke is 9. See, radial spoke is 9. Some of you are confused with the linkers. Linker is 10 and radial spoke is 9. 9 in both the case, right? 9 in both the case. So this you can take a screenshot or note it down. Main difference between 9+2 arrangement and 9+0 arrangement. If you want, you can take a screenshot here. So let's move ahead. So this is clear to all? No doubt. Just give me a thumbs up if you have understood this 9+0 and 9. See, why I am taking some time to explain this, because first you are reading it for the first time. Till now you haven't heard about 9+0, 9+2. And many students have the confusion in these both arrangement, right? Okay, great. So if you have understood, just give me a thumbs up so that I can move ahead. Or if you want me to explain any part of it, just let me know. So now, one more thing before going to other topic. If I say, so this is statement. Now again, this is a star question. So let me see how many of you can answer. This is a 5-star question. And true false, right? It is a true false question. Let's see how many of you can answer it. So here, statement says that 9+2 arrangement is present in flagella of bacteria. Now think it very, very carefully and then answer. You have to answer true or false. 9+2 arrangement is present in the flagella of bacteria. True or false? Think before answering. Let's see. Those who are chatting other than answer, don't do that. See, I am trying to trick you. And you are falling into my trick because most of you are writing true. See, 9+2 arrangement. What I have said, this was a critical question. 9+2 arrangement. It is the arrangement of microtubules. Isn't it? Now, if I say this statement, microtubule is present in bacteria, what you will say? Microtubule is present in bacteria. True or false? True or false? Now, for this, let's say this is A and this is B. Tell me, Statement B is true or false? Jenny is telling false. Okay. Venkata is telling false. Sekhar is telling false. Okay. So microtubule are present in bacteria. This is totally wrong. This is false. Because microtubule is not present in bacteria. If microtubule is not present in bacteria, how can 9 plus 2 arrangement is present? So remember, 9 plus 2 or 9 plus 0 arrangement is not present in bacteria or prokaryote. It is only present in eukaryote, right? So it is not in, so this will also be false. So you can note it down. 9 plus 2 or 9 plus 0 arrangement is only present in eukaryotic cell, not in prokaryotic cell. Is this point clear? That's why in the beginning I said this statement. Let's see. I'm writing this statement. Flagella is, this is a statement based question. See, this is also a type of question which is very commonly asked in NEET. So today I am giving this question on that basis. Let's say first, statement 1, it says flagella is present in both eukaryotes and bacteria. Second statement, it says that flagella in prokaryotes have different protein as compared to eukaryotes. Now options are, I will give you option also. So option number A is both 1 and 2 wrong. Option B says both 1 and 2 is correct. Option number C says that 1 is correct, 2 is wrong. Statement 2 is wrong. And the fourth, D, 2 is correct and 1 is wrong. Tell me what is the correct answer here. Which option will you go for it? Which option you, okay, already many of you are answering. Pooja, Ranga, okay, Geeta, Khalil, Lashya, yes, Anivesh, Reshma, very good. So all of you, I think you have understood the point because all of you are telling option number B. So flagella is present in both prokaryote and eukaryote. Totally true. But flagella in prokaryote have different protein as compared to eukaryote. So this is also right. So it is both statement 1 and 2 is correct. Understood this point? So I think this point is crystal clear. Now let's move ahead. So now it is the quiz time. So quiz time, so we will put the timer on so that you do it fast. And you have, see, first question. So let's complete it in within 50 seconds. First question says that centriole are made up of. What it is made up of? Is it made up of DNA? Is it made up of protein filament? Or is it made up of microtubule? Or is it made up of ribosomes? Tell me the answer. For the first, the correct answer will be, I'm getting C, very good. Geeta, Alia, Harshit, Rani, Neerath, Sanmukh, Utkarsh, Lakshya, Lovely, very good. Khalil, Pinky, very good. So you are telling microtubule, right? We have just seen. And what is the arrangement? So just seen the centriole has a 9+0 arrangement. Very good. Now what type of structure do centriole have? Again simple. Just I said 9+2. So what you will say? Centriole. So centriole has 9+0. And here this 9 is what? Okay, let me see how many of you can answer this. A. Okay. Harshit is telling A. Sai is telling A. Alia, Geeta, Ganesh, Srinivas, Faz. Very good. So very good. You all are telling A. So 9 triplet of microtubule is present and these are peripheral. Peripheral. So here the correct answer will be triplet of microtubule. Now the main function of centrosome is, what is the main function? Function of centrosome. Remember, I have drawn a diagram and I have told that we will study it in the next chapter. And what is the name of the next chapter? Cell division, right? So yes, let's see how many of you can answer this. I have already given you a hint. I have already given you the hint. Okay, Geeta correct. Khalil correct. Sai correct. Vijay, very good. Vijay Kumar, Chandan, Tulsi, Lashya, very good. So it is simple B. B is correct answer here. Then next one. The centrosome is made up of how many centriole? Again, very simple question. Centrosome is made up of 2 centriole and they are perpendicular to each other. They are perpendicular to each other. Very good. So now let's move ahead. Now this is another question. Now here all of you have to answer it. This is review of what we have studied till now. Now what you have to, how you have to answer here? You have to just write. You have to just write A. What is A? For example, let's say powerhouse of the cell. So if it is, let's say lysosome, so you have to write A lysosome. Then only I will understand. Right? Fine. So already you are started answering. Very good. All of you are answering mitochondria. So powerhouse of the cell is mitochondria. Of course, you must have studied earlier also that the powerhouse of the cell is mitochondria. Now next one. Next one. Middleman of the cell. Now this one is tricky. Although I have explained. Middleman of the cell. Remember, traffic police of the cell, postman of the cell, middleman of the cell. Very good. Venkata has given the answer. Very good, Venkata. Sheikh has given correct answer. Neerath, Sai has given correct answer. Tanish, very good. Okay, so here middleman of the cell, we have talked about Golgi bodies, right? Golgi body or Golgi apparatus. So Golgi body is called the middleman of the cell. Why? Because it take protein, lipid from the ER and then it transport it inside or outside the cell. It do the packaging, so also called postman of the cell. Then protein factory of the cell. Protein factory of the cell. Very good. Sheikh has said ribosome. Tanish, ribosome. Okay. Protein factory of the cell is called ribosome. Now here one more thing that many of you are also writing, rough endoplasmic reticulum. See, ribosome present on rough endoplasmic reticulum. That's why rough endoplasmic reticulum is forming protein. So the function of formation of protein, if you have ribosome in the option, always go with ribosome, right? So protein factory of the cell is ribosome. Then kitchen of the cell, simple, very good. You all are already answering chloroplast, very good. So it is chloroplast because it prepare food. Chloroplast is the kitchen of the cell. Suicidal bag of the cell. Suicidal bag of the cell. Suicidal bag of the cell. Neeraj has given the answer. Very good, Neeraj. Neerath has also given answer. Okay, chat is moving very, very fast, so I am unable to read most of your names. But yes, you all are answering correct. It is lysosome, right? It is lysosome. It is lysosome. Then last question. Again, this I have already asked 3-4 times, but again I will ask you this. Very good. Reddy has given answer. Right. So yes, this we have already done several times, so it should not take time. So it is mycoplasma. It is called the jokers of microbiology. So in this way also question is asked in NEET. They will simply give you this statement and then you are given different option, and or you may be given match the following based on these options. So well done. You all are doing excellent. So for that I will give you all stars, right? Because you all are responding well. You are not those lazy kids who are not responding and not answering. So it's very good. So now let's move ahead. You all are learning a lot of concept, and trust me, when you will go back in your regular classes will start. All of you who are attending this class will be far ahead of all those who will start during the class. So use these classes very nicely. Now next cell organelle is nucleus, right? So very basic part of the nucleus, let's consider. So if I say nucleus, what are the different part of nucleus? So first is, there is nuclear envelope. Nuclear envelope. So how does a nuclear envelope look like? So I will draw a big diagram for you here so that you understand it well. See, this is a nucleus, right? If you see very carefully, this is called the outer membrane of nucleus. Outer membrane. And then this will be called as inner membrane. Inner membrane. Then in between the outer and inner is present the intermembrane space, or we also call it perinuclear space, right? We also call it perinuclear space. Perinuclear space. Now, so can you all recall, we have seen some other double membrane bound cell organelle which has outer and inner membrane. What are those? Can you tell me? Mitochondria and chloroplast. So now remember, there are 3 cell organelle which are double membrane bound. What are the 3 cell organelle? They are mitochondria, chloroplast and nucleus, right? So nuclear envelope consists of outer and inner membrane. Now if I say, you can clearly see this nuclear envelope is not regular. At some places, this outer and inner membrane are coming and joining with each other, and at that place there is a pore which is formed, and that is called nuclear pore. This is the nuclear pore. Now let's get inside the nucleus. Inside the nucleus, the fluid is called nucleoplasm. It is called nucleoplasm. Is it clear? Nucleoplasm inside the nucleus. Then there is a dense spherical structure, right? There is a dense spherical structure, and that dense structure is called nucleolus, right? As you can see here, nucleolus, dense structure. Then further inside is small thread-like structure, and this thread-like structure is called chromatin. It is called chromatin. So overall, now let's see different parts. I will again repeat. Nucleus has outer nuclear envelope, as you can see here. Is nuclear envelope regular? Answer is no. It has pores in it. So at the pore, what happened? Outer and inner membrane join with each other. Inside the nuclear pore, inside the nuclear envelope is the nucleoplasm. Inside the nucleoplasm is a dense structure called nucleolus and thread-like structure called chromatin. So in the next class, we will study all these structures one by one, right? What is the role of nucleolus? How is chromatin formed? What is the different composition of chromatin? What role does the nuclear pore play? All this we will do in the next class. So go back today, read NCERT, go through the diagrams given in this chapter, solve some MCQs, and you can also rewind and see this video lecture again. So here I will stop the class for today. Thank you all of you for participating in the class, and you are learning a lot. Thank you.